We're very excited today to have Dr. Matthias Hebrok with us. The topic of the talk is New Regulators of Human Beta Cell Function. And just a little bio for him. Dr. Hebrok is the professor and chair of Applied Stem Cell and Organoid Systems and serves as the executive director for the Center for Organoid Systems, COS, at the Technical University of Munich, otherwise known as TUM. And he also directs the Institute for Diabetes and Organoid Technology, IDOT, at Helmholtz, Munich. And he received his diploma degree in cell biology from the Albert Ludwigs University in Freiburg, Germany, performed his doctoral thesis at Max Planck for immunobiology, and conducted his postdoctoral research at HHMI at Harvard. He held the Herbert Johnson Distinguished Professorship in Diabetes Research until 2022, and served as the director of the Diabetes Center at UCSF from 2010 to 2020. This group has made very important contributions to the field of pancreas organ development, stem cell biology, and regenerative medicine. This work generated sophisticated human stem cell tools to build functional replicas of human hormone-produces islet structure, a novel cutting-edge technology enabling cell therapy for patients with diabetes. And he has also advised prominent academic diabetes centers, served on the SAB of several biotech and stem cell companies, and co-founded Minutia, which is a company developing novel nanosensors to monitor activities of stem cell-derived transplants in patients. Welcome, Matthias, and we're very interested to hear about some new papers coming out of your laboratory.
Great. Thanks a lot, Monica. Yes, thanks for inviting me. It's great honor to be here and talk to you about our work. As you just stated, I would like to talk about two papers. Actually, one is out already, and just going to summarize this, and the other one hopefully is going to be out very, very soon, essentially imminently.
Right.
Okay, let's see here. Let's hope this works. There we go. Okay, great. My university asked me to just put this up very quickly.
Sure.
So what I would like to do is I would like to tell you a little bit about the work that we've been doing with human pluripotent stem cells mostly. And we have been studying this in the group for many, many years now, actually, a decade and a half or probably even longer. And the idea, of course, is that you can take stem cells and you can follow the differentiation that the embryo has shown us to go through the different steps and end up which we call enriched beta cell clusters, or in short, EBCs. And I just want to point out that I think these applications for these cells are multifold and quite interesting. I don't have time to go through all of those, but essentially you can look at human versus rodent beta cells, which is important because they're very similar but not identical. We do a lot right now on designing cells, trying to essentially make those better, more functional for cell therapy, but also for drug screening purposes. Then, of course, there's the application of cell therapy using potentially even patient-specific therapies. Then there's the microfluidics, which I, again, don't have time to talk about today. And then we have an interest in pancreatic cancer. Certainly is not what I'm going to mention today. What I would like to do today is talk a little bit first about diabetes susceptibility and essentially using this technology to get a better handle on understanding how these genes function. Just want to start out by saying that there are many GWAS studies that have been done to identify loci specific for type 2 diabetes. And this study that was published not too long ago essentially identified in this particular case more than 318 or so of these loci. And we had a long-standing and really fruitful and actually fun collaboration with Al Nadi and Mark Keller and their group. And what those guys did is they essentially generated diversity outbred mice by crossing 8 different strains together. And then they used those to isolate the islets, do an analysis of the islets, and looked for specific parameters, including, for example, insulin secretion or perturbed insulin secretion. And then we're using mouse GWAS and comparing this to human GWAS to identify molecules that potentially were missed in the human situation, but that they couldn't find in the mouse. And one of the molecules that they found, published first actually in 2019, but in a follow-up paper then by Chris Empfinger in JCI Insights a year or so ago, this molecule is a transcription factor called ZFP148. That's the mouse version, the rodent name. The human name is ZNF148. And this molecular transcription factor actually impairs insulin secretion in rodents. So there is actually a connection to the human gene that is associated with fasting insulin, glucose, and HOMA-RI, but surely the function and what this gene is doing in human beta cells was not known. And that's how we, in our collaboration, got interested in this. And at that point, a very intelligent postdoc, Eleanor de Klerk, she's now a scientist at RNA Therapeutics, she decided to take this project and try to understand what ZNF140A potentially does in human ES cells and in beta cells derived from these ES cells. So the first experiment she did is she just followed the strategies well established in the lab. Essentially, she went in and generated a number of different lines in which she has eliminated ZNF already in ES cells. And then, as you can show here by transcriptome expression, the expression of those knockout lines is down. She went on to generate EBCs, or these enriched beta cell clusters. You see in green here the expression of insulin, and in the control, the expression of ZNF148. So it's robustly expressed in our stem cell-derived beta cells. And then as a control, of course, she looked for the expression of ZNF148 in the knockout lines. And as you can see here, both these are two knockout lines. The expression is completely, as we expected it to be, is completely lacking. So the tool works. We can essentially eliminate 148, and we can, as you can see here, still go through the differentiation to generate the ES cell-derived beta cells. And that is again shown over here. So these are the controls shown on the top. These are how the clusters look. This is in a bright field. Same here in the elimination of ZNF148. And we're looking because our line has an insulin GFP built in, which means the GFP is in a control of the insulin promoter. And as you can see, the clusters that form have high level expression of ZNF, of GFP. Just we can go and, of course, do much more quantitative analysis, which we do with FACS analysis. But essentially, the take-home message is here. If you look at the expression of C-peptide in the control situation versus the knockout concentration, then the population of cells that are C-peptide or insulin positive is essentially unperturbed. So this was all good and fine. But then we asked the question, does the elimination have a specific...
Quick question from the chat. I wanted to get to it while we're still on the slide. From
Sure.
Eric Schomakers, the Biomea Fusion, or BMEA, is currently clinically developing a drug candidate, BMF219, that seems to be triggering the differentiation of stem cells into novel beta cells as a strategy. BMF219 impacts the menin signaling pathway. You have previously published about this. Any thoughts about their strategy? Initial results look very promising.
Menin, of course, has to do with the proliferation of cells. Mutations in menin are known to give rise to insulinomas. That's something that has been studied in great detail, not by us, but by others. So I'm not exactly sure at what point this molecule has an effect on the differentiation. We have an interest in menin, we have an interest in insulinomas, but we have not tested this molecule, so I can't say anything specific about this one.
Okay, thanks for taking that.
Okay, good. So getting back to that, so the first thing we did is we did a glucose-stimulated insulin secretion. So essentially what we're trying to do is we're trying to understand if our cells can respond to glucose challenge. And then we've looked this in a control, in comparison to the knockout of the ZNF148. And actually there was an increase in glucose-stimulated insulin secretion. That was exciting because, as I mentioned to you before in the first slide, that in rodents, actually, ZNF is a repressor of insulin secretion. So we had a similar phenotype here. And then we essentially dug a little bit deeper. And as it turned out, what was interesting is that if you do a dynamic expression, or dynamic glucose-stimulated insulin secretion, you see that after the first peak, actually in those mutants that are shown here in this light blue, you see that there's higher levels of insulin secretion compared to the controls. So they seem to be secreting insulin for a longer period of time. And they also had an increase in the timing of insulin secretion when you give potassium chloride. So there's something that in the mutants changed the way insulin secretion happens. So we went forward and did an analysis, an RNA-seq analysis, tried to understand which kind of transcription factors or molecules are changed in the context of the mutant. And what we found was that calcium ion binding was the highest Go term that came out. And so that's interesting too, because of course we know that I just mentioned to you that there seems to be something increased in the insulin secretion. We know that calcium is involved in insulin secretion in the whole process. Then furthermore, if you just look at specific genes that were changed in the expression, we found that a number of those that had to do with cytoskeleton remodeling, but also calcium flux, as I just mentioned to you, but also vesicle exocytosis were changed. In particular, this molecule called ANGSA1. ANGSA1 is one of the members of a family called annexins, and these are quite interesting members of a family that is, in particular case for annexin 1, co-secreted with insulin in response to glucose that has been shown in rats before. It's also expressed in most of the insulin-containing granules, and annexin 1 treatment was associated with the movement of the reserve pool of insulin vesicles in beta cells. So we got really interested in this particular molecule, ANGSA, but not just ANGSA1, as I show you in a second, but the whole family. So the idea would be that we have the insulin vesicle, and we have essentially the requirement of this vesicle to be attaching to the membrane. And our idea would be that, as has been shown before, that ANGSA2 or ANGSA1 plays an important role in this. And the reason why I'm talking about ANGSA2 rather than ANGSA1 is because if you look at the expression in human cells, as was published before by Sandberg et al., you can see that the expression of ANGSA2 is highest in the proportion of the human beta cells. So we focus on ANGSA2 mostly. We've looked at the other ones as well. Again, I'll show you the publication, the citation later on, so you can look it up. But all of those, ANGSA1 and ANGSA2, as well as ANGSA3, are upregulated upon elimination of this transcription factors ZNF148. So what we did therefore is we asked the question, how does ANGSA2 expression potentially is changed? Because it is known that these molecules not only bind to vesicles, but that they actually can translocate from the nuclear region to the membrane. And if you look at our controls, our beta cells that we were generating, you see that the expression of ANGSA2 here in particular is actually located to the nucleus. That's not necessarily the place where it's supposed to be. But in the ZNF mutants, you can now see that the location of the protein changes dramatically. It goes out of the nucleus and actually goes outside to the membrane. And this is shown here in a higher magnification picture, just focusing on a few cells, that now the expression is not in the nucleus any longer, but it is on the outside of the membrane, where, of course, the insulin vesicles would be located prior to secretion. So that is also the case for ANGSA2 in normal human pancreas. You also see here that the expression is not in the nucleus, but is also in the normal pancreas localized to the cell membrane. So essentially, by eliminating this transcription factor in our stem cell-derived beta cells, we were generating a situation that is more closely related to what we find in a pristine beta cell in our body. So the idea then therefore is, as I mentioned to you before, that ANGSA is involved in the vesicle transport of insulin somehow towards the membrane. But the story, of course, as it always is a little bit more complex, because actually ANGSA2 does not exist as a homodimer, but actually as a heterotrimer. And what it needs and what is required are these molecules of the S100 family. And these molecules bind to ANGSA, and essentially they form the bridge between these two of these ANGSA molecules. And that seems to be the bridge that allows for the vesicle to come to the membrane. And so we therefore asked a very simple question: if that is the case, and S100 molecules seem to be required for this, are they somehow also regulated by this factor ZNF that we are interested in? And the answer was yes. So you can see here, we looked at a number of these S100 factors, and the expression is up in a number of those in the ZNF148 knockout lines. Particularly, we were interested in S100A16, and as you can see here, again, the expression is increased, which means that ZNF under normal conditions works as a repressor. So we asked the question, can it directly bind to the promoter elements of S100A16? And the answer was yes. We found a consensus binding site for ZNF148 in the regulatory elements of the gene. And then by performing a chromatin IP, we were able to show these are controls, gastrin controls, that there is binding, direct binding of ZNF, of the transcription factor, to the regulatory elements of this molecule S100A16. So therefore, the hypothesis would be that ZNF acts as a repressor and does not allow for the sufficient deactivation and the expression of this molecule S100. And in the absence of ZNF148, S100 is expressed, and S100 then binds together with ANGSA2 and allows for the appropriate secretion of insulin vesicles. That, of course, leads to the next question, and the next question would be, can S100A expression mimic the ZNF148 loss? So can we overexpress S100 and therefore rescue the phenotype? And so we tried to do this. And this shows you the power of the system, of the stem cell system, because not only can you eliminate genes, but you can also, at a time of our choosing, overexpress specific molecules. And in our case, we're trying to overexpress S100. We also had to ectopically give ANGSA2 to see an effect. And so this is the experiment that I'm going to show in a second. These pictures I've shown you before. This is the overexpression, but there is no doxycycline given. So this is just a line. It doesn't overexpress it. ANGSA2 is positive or is given, is applied as a protein, but it still localizes to the nucleus, as I showed you before. And now if you overexpress S100A16, giving doxycycline, which does that, together with ANGSA2, now you see again the change of the protein localization away from the nucleus towards the membrane of the cells. So there is a little bit of a rescue. But then, of course, the real question is, can you not only rescue the location, but also does it have an effect, a physiological effect in terms of glucose-stimulated insulin secretion? And this is a little bit of a busy graph here. I apologize for that. But I hope you can see that essentially those lines in which we have both activation, the expression of S100 and giving the ANGSA2 or ANGSA1. So it's not specific only to this molecule or this member of the family, but it also can work with A1 as well. And in both of those cases, the red and the green indicates that there's higher secretion of insulin upon glucose stimulation. And that is just shown over here. The effect is not dramatic, but is statistically significant. As you can see that the red as well as the green have higher levels, higher area under the curve compared to the controls. So the answer is yes, by just providing ANGSA proteins, but also overexpressing S100, can we rescue the phenotype. Now, does this have any connection to the human situation? We think that is the case as well, because if you essentially use a knockdown of ZNF148 in human islets and then look at the glucose-stimulated insulin secretion here in a stepwise manner, you can see that the gray line, which is the knockdown of ZNF, performs better than the controls, the normal beta cells, without reduction of ZNF148. So even in the context of a pristine human beta cell, human islets, you see an increase in insulin secretion upon reduction of ZNF, as shown over here. So this was the first part of what I wanted to share with you. And as I mentioned to you before, this was published earlier. Eleanor was the first author here. It was published in JCI Insight in 2023, just a few months ago. And the idea is over here that in a beta cell, you have insulin secretion. You don't have the perfect alignment of the ANGSA molecules together with S100A. But if you reduce or eliminate ZNF148, then you have essentially the formation of this bridge, and therefore you have activation and rapid transport of these vesicles to the membrane and an increase in insulin secretion. with that, I would like to essentially move on and tell you a second story that hopefully, as I said, will be published any day now. And that has to do with what I would call an old friend, because my lab has actually worked on this factor. It's another DNA binding protein called SOX9 for more than a decade again, but mostly actually in the context of deregulation of beta cell function or in the context of the pancreatic cancer. And just to orient you, SOX9 is a transcription factor or a DNA binding protein that is known to be expressed in pancreas progenitor cells, shown over here. And the expression actually stays high in the pancreatic duct cells. It's reduced in the acinar cells, and it was also at least believed to be completely lost in the cells that would move down towards the endocrine lineage, be it the beta cells or the alpha cells. Now we have evidence that actually there is a residual expression of SOX9 in mature beta cells, albeit at very, very low levels. And that's reminiscent of other factors, for example, neogenin-3, that also is expressed at high levels in the endocrine progenitor cells, then becomes significantly downregulated, but still has a function in the mature beta cells. So there is evidence from other factors that this can exist. So what is the evidence that we now think that SOX9 actually not only is expressed in mature beta cells, but actually has a functional role? And we've done a tremendous amount of work in this. But before I start out, I should just point out that the expression of SOX9 actually can be found, as shown over here in the duct cells, but also in some of the beta cells at much lower levels, shown over here. Furthermore, if you look at this and just focus on the expression in the beta cells, you can see this that in human beta cells, there is expression of SOX9 as well. So we started out, and this took us a long time, because we had to convince ourselves that this really is expressed and also has a function there. So we used a number of different lines first, and actually focusing on rodents in the beginning. And we used a number of different models. The insulin Cree is the one that I'm going to describe to you, but we also used a MIP-Cree to be able to eliminate SOX9 specifically in the beta cells after they are formed, as well as a different model in which we just took out islets and then transfected with a virus to eliminate essentially ex vivo the expression of SOX9 as well, and therefore to ensure that there were no effects that are coming from somewhere else in the body. So let me just give you a few of the experiments that we've done in this one model, the insulin Cree, which again, we have most of the data. So the first thing we did is trying to convince ourselves that SOX9 is expressed in beta cells. And with the antibodies that we have available that work quite well, as I should show you later on in other cell types, particularly in the duct cells, we were not able to see this. So the level is quite low. So we had to use amplification methods, and we used an RNA scope method here. And I think you can clearly see that the red signal, which is the SOX9 signal, is clearly in cells that are also insulin positive, as stained for in green by insulin. The reason why we believe that is it's not only in the islets, as I can show you here, but also in these structures where it is known to be expressed at high level, and that's the duct structure. Furthermore, because we can do this in the mouse in a genetic system, we use the insulin Cree to eliminate SOX9, and now you see that in the beta cells, again, the signal has disappeared. So SOX9 now is not present anymore, as it shouldn't be, because the insulin promoter, of course, is active in beta cells. Now, in contrast, you still see activity, as you should, in the duct cells in which the insulin promoter is not active. So we do believe, again, as a take home here, that SOX9 is expressed in beta cells, but is expressed at low levels. Again, we did a lot of experiments with those mice, and I only show you a few because I'm out of time here. But when we actually do the experiment in looking for glucose levels upon stimulation, then you can see that upon stress, the insulin Cree SOX9 mutant mice have higher levels of glucose. And it goes in a stepwise fashion. These are the heterozygous and these are the controls. So their insulin, the properties to regulate glucose levels with insulin is perturbed if you eliminate SOX9 in the beta cells. Actually, the circulating insulin levels are increased, but if you look at glucose-stimulated insulin secretion, you can see that those cells are not able to start a robust response in increasing it from the unstimulated towards the stimulated, which is different in the controls shown over here. And therefore, the stim index of glucose-stimulated insulin secretion is lower in those mice. If you look at the islets and just start to take a look at the expression of insulin, you can see already that it's patchy. It's not as robust, not as strong as you would see it in the controls. And that is not only true for insulin, GLUT2, the transporter, glucose transporter, also is perturbed, as well as there seemed to be an increase in proinsulin. So the maturation of insulin seems to be perturbed in those mice as well. So therefore, let me just summarize that part very quickly. The loss of SOX9 in beta cells leads to glucose intolerance in mice. The reduction in SOX9 impairs adult beta cell appearance and function. To get to the mechanism of how this might work, we performed an RNA-seq experiment, and there we had a little bit of a surprise. And the surprise was that almost nothing changed. We had here only 196 or so genes that were upregulated, and there was very little that essentially we could put our handle on in terms of any kind of specific process. So if you look at the gene ontology or the CAC pathway analysis, there's nothing really where you would say this makes perfect sense from what we know about SOX9 and how it potentially affects the beta cell functionality. So we were a little bit stumped at that point. But then we got lucky because Vasilis Nitrantis joined the UCSF Diabetes Center at that point. He's a bioinformatician. And Vasilis actually, during his postdoc, had worked on using bioinformatics tools to look at alternative splicing. And by using our data sets that we had generated, he took a look at this, and now we found something that was exciting, and that is that mRNA splicing, regulation of alternative splicing, spliceosome, as well as, again, the spliceosome here in the CAC pathway came up. So that clearly indicated that upon elimination of SOX9 in those islets, again, these are still rodent islets, there was a change in the alternative splicing in those cells. And so then, again, using his analysis, looking at alternative splicing, spliced variants, he found that the factor that actually was the most changed is called SASF5. So why is this important that SASF5 is changed? And just to point out here, we would not have found this by just looking at the gene expression set, because it is a little bit reduced, but it is not significant. So what does SASF5 do? As it turns out, SASF5 belongs to a member of molecules that are enhancers of splicing. They bind towards the exon as well as in the intron as well, and they are critically involved in the splicing process itself. So SOX9, therefore, seem to have an effect on the splicing of enhancers that regulate alternative splicing. Let me just give you a primer of how alternative splicing or splicing in general works. We have a number, or most genes, not all, but most of the genes have a number of different exons, and it's therefore possible to essentially, in a modular fashion, change the structure of the protein by combining different exons. So in this particular case here, for example, we have the yellow exon present. In this particular case, the yellow exon has been removed, and then the purple one is not included. So you can get a protein 1 or a protein 2 out of it, and they, of course, can have different functions depending on what kind of sequence is included or eliminated. But there's something else that splicing can do, and that is that there is what we will call a poison exon that is included. And by this inclusion of this poison exon, it actually leads to an early termination, and therefore no protein or no functional protein is translated. And that is exactly what is also present in SASF5. So there is a poison cassette exon that is present in the gene in SASF5, and to obtain a functional protein, it needs to be eliminated. If it is included, it leads to premature termination, a codon that leads to premature termination of translation. So what we found is, and it was quite remarkable, is that in the insulin-free SOX9 minus islets or beta cells, we do have the inclusion of this premature poison exon. You can see that the expression here is significantly higher compared to the controls. So there are a few of those transcripts that occur in the control as well, but it is much, much higher in the mutant lines. And vice versa, if you look at those versions of the protein that generate a functional protein that have this poison exon eliminated, you see it is significantly higher in the controls versus the mutants. So the mutants, therefore, for whatever reason, seem to be leading to, at least in this particular case here, the inclusion of this poison exon. And you can go through all of the different versions that are known for SRSF5. Many of them are protein coding. Several of them have the retained intron. And really excitedly, you can see that in the controls, you have higher level expression of those transcripts that will give rise to a protein coding sequences, versus in the mutants, the protein sequences, or those that will give rise to proteins, are reduced. And vice versa, if you look at those transcripts that will not give rise to a protein, those are increased upon elimination of SOX9. So SOX9 elimination has a profound effect on the presence of a protein that is required for alternative splicing. And we cannot only look at this at the level of transcriptome. If you look at the protein itself, you see SRSF5 to be expressed at significant levels here in the nuclei of beta cells. And in the mice in which we have eliminated SOX9, you can see that the protein is more diffuse and is not expressed at higher levels in the nucleus any longer. So the second summary then, therefore, is that SOX9 affects beta cell functions in part via modifying alternative splicing. And SOX9 appears to be at the apex as it deregulates alternative splicing of essential spliceosome enhancer factors. Of course, towards the end, I would like to come back to our system and ask the simple question, has this anything to do with human beta cell function? I just, again, want to show this slide that I already shown before. There is low-level expression of SOX9 also in human beta cells. And now, as I demonstrated this before, we can use genetic tools and CRISPR technology to eliminate SOX9 at a time of our choosing. And so we're doing this in this particular case by expressing guide RNAs against SOX9. And these are a number of lines that we have generated. This is the minus DOX control. This is the positive control. And as you can see here that upon giving doxycycline, what you see is that there's a reduction in the message, and this is quantitated down here. So this is different in the lines I described to you before for this different transcription factor ZNF, because we're doing this now in a temporal manner. We're not generating an ES cell line in which SOX9 has been eliminated. We're doing this at the stage when the beta cells are already starting to form. And it works, as I've shown you here, in real time also for a protein. So this is at the early stage at day 11, when we know that there are progenitors, pancreatic progenitors that are forming that are really highly positive for SOX9. This is the no DOX control. And as soon as we give DOX, you can see that there's a very significant reduction in the SOX9 protein, not just the message, but the protein. Because we do know that SOX9 is required for the formation of pancreatic progenitors and plays a really critical role in here, we are only giving doxycycline treatment after the progenitors have formed towards the mature beta cells. And if you do that, then you can see we get to the point where you actually have the production of insulin-positive cells. So, but these insulin-positive cells, by doing single-cell analysis, are changed compared to the controls. So these are the control here. This is the no DOX treatment. So SOX9 is expressed at the levels that it normally would be. This is the DOX treatment where we have the reduction of SOX9. And as you can see, there is a reduction in the population of these cell types. These are the ones that would correspond to the more mature beta cells, versus there's an increase in the population of those cells over here, and those would correspond to the more immature progenitor cells in the beta cell lineage. And that also has consequences in terms of the physiology of these cells. So now we are comparing here the DOX treatment in red. So the gray is the control, and you have glucostimulated insulin secretion. There's a response as we see it. And then if you have DOX treatment, you see this glucostimulated insulin secretion is reduced. So therefore, we have an impairment in those cells that have lost SOX9 towards the later stages of differentiation in the beta cells in terms of their functional properties. And this is just the quantification of this, where you see the area under the curve is reduced. So then, of course, the question is the mechanism similar to what we were able to find in the rodent beta cells? So in other words, does SOX9 regulate alternative splicing in human beta cells? And the data that we have indicate that this is the case as well. So there is a homolog, it's called SSF5 as well, that is similar to the one that I described to you in the rodents. And similarly to what I described there, we also see here that there are retained introns that lead to non-sense-mediated decay, as well as another protein formation. And those, upon essentially elimination of SOX9, have higher preponderance compared to the controls. And vice versa, those versions that result in protein coding splicing events, these are shown over here, those are the ones that are reduced upon reduction of SOX9. So similarly to what I described to you in much greater detail for the rodent situation, we also believe that in human beta cells, SOX9 has a similar role. It affects alternative splicing, and it does so like it did in the rodents on the level of these enhancer proteins that are critical for alternative splicing. So therefore, I'd like to conclude and showing you just one more little bit of data, and that is, what is the mechanism behind of this? It actually is known that SOX9 can modify splicing. It's just, but it has only been shown in one cancer cell line to our knowledge. And there is an association with another protein. It's called Y14. This is an RNA binding protein that is involved in binding to the exon junction complex. So essentially, this molecule Y14 over here is in complex with a different protein called MAGO, and they bind to these proteins that have been undergoing splicing, and then they seem to be also involved in the export as well as in the capping of the RNA structures. So the question, therefore, is, as it was shown or indicated in these cancer cells, in our beta cells, is there a connection between SOX9 and Y14? And to do that, what we did is we looked for a proximity assay to look for direct protein-protein interactions. And the idea behind this is that you have essentially two proteins that are connected, and they can be bound by antibodies. These antibodies have a tail that forms a circular structure, and this circular structure can be amplified as concatenamers, which means they are still retained and have the connection to the location. And then these can be used to, they have consensus sequences for probes that have a fluorescent protein. So essentially, when you see a signal coming out, you can see where it is, and it would indicate that these two proteins are closely together. And so we've done this for Y14 alone. You see the proximity ligation assay, there's nothing here. SOX9 alone, there's nothing here. But if you put together Y14 and SOX9, you see a robust signal that is forming in those cells in the right place in the nucleus. So we therefore think that yes, as in colon cancer, SOX9 can bind directly to Y14. Although exactly how it affects splicing, that's something that still needs to be shown. With that, I would like to conclude. SOX9 is a novel regulator of mature beta cell function. SOX9 affects beta cell function in part via modification or modifying of alternative splicing events. SOX9 regulates alternative splicing of essential spliceosome enhancer factors, and SOX9 interacts with Y14. With that, I would like to come back to my first slide and hopefully have convinced you that the system that, of course, not just my lab, but many, many other labs have developed as well, is a really great system to not only just look into human beta cell function, but also to perturb it, to learn something about molecules or genes that have an effect in the disease. And then, of course, and that is the idea, to use all that information to make these cells better, to design cells that, for example, are self-healing, that are just more robust and can be used then for cell therapy purposes. With that, I would like to show the lab. I think I went through. I forgot to mention that the second part had the picture up, was done by Sapna Puri, who is now at Minutia, vice president there for cell biology. And Sapna did this for many years, did a superb job in putting this together. She did, towards the end, quite a bit of help from another postdoc, Hasna Machi, shown over here, now in Munich, who essentially is taking up the next steps of this alternative splicing project. And with that, I would like to thank you for your attention.
Great. Really just an excellent talk. We have so many, I've been fielding so many comments to that end in the chat. So many people are really enjoying the talk.
Thank you.
A lot of accolades. There's a couple questions. I didn't know, I let them pile up here, and I wanted to, I can ask a few. And also, let's see, let's start with Leonardo Ferreira.
That is not a surprise. Hi, Leo. How are you doing? You have to unmute yourself.
Hi, yes, sorry, it's a bit slow. Yeah, nice seeing you as well. I just have maybe a very broad question. Really fascinating work where you find these transcription factors, the ZNF and the SOX9, which are not necessarily, 10 times more expressed in the cells of interest, but it's still extremely important. And especially the ZNF factors, sometimes I run into them as well in RNA-seq data. So do you have a sense of, what the fold change should be and how to go about this? So this is a very general question, but it's really interesting to me how to find
there's lots of CNF factors, right?
Yeah.
This is a particular one. It actually, I think, if I go back, I think it's expressed in immune cells as well, but don't hold me to this. We have looked, I didn't have time to show you, but in the paper, we have looked at how the expression of CNF changes over time in beta cells and stem cell-derived beta cells. It's robustly expressed. So I don't know if that answers your question, though.
Yeah, I think in the end, it's like sometimes maybe good to have different sources of data to show this gene is really expressed by RNA-seq, by qPCR, by protein.
Yeah, and we can see it by protein. So ZNF is clear there. SOX9 is very, very different. It's expressed at very low levels. So we had to go through really extreme lengths to be able to show it. But again, I only showed you the highlights of the study. We've done a number of different mouse mutants as well, isolated islets, and we always see similar phenotypes that it actually has an effect on the physiology of the beta cell.
Yeah, thank you.
Great. If anyone else is interested in asking a question live, please raise your hand and we'll unmute you. Let's see. Here's another one. Kati Girda from Boston. Nice work. Fantastic talk. Did you determine the removal of positive exon in SOX9 --, improves glucose tolerance, IS?
I'm not sure if I understand the question.
I can clarify the question.
Yes.
I had, I think, two questions in the chat. One was regarding the first talk, where you have shown GNF148 knockout from the islet. We have studied the human islet. The question was regarding the insulin, first phase insulin secretion usually abrupt in type 2 islet, right? So did you determine similar effect in type 2? If didn't, would you expect the same?
That's a good question. You mean essentially removing ZNF in type 2 islets? No, we haven't done that. That's something we didn't have access to those islets. But that's a great question to ask. Yes. My guess would be it would work as well, particularly if this would be in the beginning stages of type 2. If you go towards the end, when essentially the beta cells is just not functioning anymore, I'm not sure if you would have a benefit out of this. But I think in the early stages, you probably would. Yes.
Thank you. The second question was regarding the second part of the talk where you mentioned the poison exon removal by the SOX9 knockout. SOX9 knockout integrate the poison exon. If the question was, if you remove that poison exon in SOX9 knockout mice, would it improve glucose tolerance or insensitivity?
So the SOX9, I don't think has this poison exon. It is in the SRSF, and actually we've looked at other SRSF factors as well. So SRSF6, and actually Dizio Ezeric has done some really beautiful work on that as well. Has had to link this to inflammation as well, and beta cell function. And this is also changed by SOX9. So I think it's a at least for some of the SRS factors, it's a common phenomenon. I cannot speculate for other factors.
Thank you.
Sure.
Sukrati, do you want to unmute?
Sure. Hello, Dr. Hebrok. Fantastic talk. And I might have missed this. You might have mentioned this earlier, but I was just curious, since SOX9 is already lowly expressed in mature beta cells. So when it comes to quantifying how much of the protein of SOX9 we need to lose in order to actually see the effects, how can we quantify that if it is already lowly expressed to begin with?
Yeah, it's a good question. It's one of the limitations. It's really very, very hard for us to do. I mean, we've tried so hard. We've tried many antibodies. We've just to see the protein, right? And we just haven't been able to do this. The RNA-scope is the only one that allowed us to limit it. So I don't know how much you have to lose to essentially have the phenotypes. That you see phenotypes when you do the elimination through the guide RNAs, and that's something that that Gopika Nair actually did when she was in the lab. She's now with Lilly. It was not a complete, it was a knockdown, it was not a complete elimination. So I do think that even a fraction, meaning 50, 60, something percent, 70% reduction is enough to give you a phenotype. But you might see different phenotypes if you have a complete elimination, which we have not been able to achieve with our strategy.
And just to kind of follow up on my previous question, are the SOX9 protein expression level comparable between the rodent and human cells, beta cells?
I don't know. The only thing I can tell you is that neither in the rodent nor in the human do we observe SOX9 expression in the beta cells using our antibodies. So therefore, the only thing I can tell you is that giving that limit, that threshold, we are under that threshold both in the human and in the rodent. That does not tell you if it is expressed at the same level. My guess would be it would be similar, but I don't have evidence for that.
Okay. Thank you so much for answering. And yes, I know Dr. Gopika Nair because I met her here at Lilly. Currently, I'm an intern here. So I had
there you go. Say hi.
Meet with her. Yes. Thank you.
Very nice. Okay, so I wondered if there's any other questions here. I see a couple of people here that might have questions, sort of like scrolling through. All right, well, I'll throw you sort of a softball question, I guess. What's the what are your next steps following the release of the secondary paper, and when might we expect to be able to access that paper?
Well, I would hope very, very soon. It is essentially it's just requiring editorial. We've had to send in a lot of this information about the single cell analysis and all this kind of stuff. Mm-hmm. I'm hoping that essentially it's imminent, and then it would come out hopefully within the next month or so. That's at least the hope.
Grab it and feature it in our weekly newsletter. So
yes, that was. Thank you very much. And then, of course, following up on that, yes, we are very interested in alternative splicing. We think that this is, and actually, Laurie Süssel just had a beautiful paper as well, and that it actually has an effect on insulin secretion as well. So I think this is just a whole new layer of course, in the beta cells of transcriptional regulation that we have not really touched. So we think that there's quite a bit there. Yes. Yeah.
No, there's a lot to be done in terms of really just even understanding still, 100 years after sort of treatment began of this disease, of understanding the mechanism of how these cells actually work.
Yes.
at the very finest levels. So, I just so appreciate your approach as a cell biologist, and now you're sort of wading into this other level of genetic control on top of that. So it's really exciting times for this new laboratory in Munich. And I also, you've got, I guess, a continued collaboration with the parent laboratory at UCSF. So just a great laboratory for those trainees who are interested in postdocs and, further exploring this area.
Thank you. Yes, no, we're really excited about this.
Yeah, and do you have postdocs positions available?
I do. The lab has grown very rapidly. I still do. We have space. So if someone is interested, please let me know.
Fantastic. Thank you so much. We'll a lot of people are saying they're actually going to reach out to you directly. So
okay.
Great to hear.
That.
That's fantastic. Can't wait to hear about what's coming next out of the Hebrok Lab. And thanks again for joining us and walking us through this really elegant work. Beautiful.
Thanks a lot, Monica.
Have a great evening. Bye-bye.
Okay. Thank you. Bye-bye.